There are multiple different V region gene segments. For simplicity, we have so far discussed the formation of a complete immunoglobulin V region sequence as though there were only a single copy of each gene segment. In fact, there are multiple copies of all the gene segments in the germline DNA. It is a random selection of just one gene segment of each type to assemble a V region that makes possible the great diversity of V regions among immunoglobulins. The numbers of functional gene segments of each type in the human genome is determined by gene cloning and sequencing. Not all the gene segments discovered are functional, as a proportion have accumulated mutations that prevent them from encoding a functional protein. These are termed pseudogenes. Because there are many V, D, and J gene segments in germline DNA, no single one is essential. This reduces the evolutionary pressure on each gene segment to remain intact and has resulted in a relatively large number of pseudogenes. Since some of these pseudogenes can undergo a rearrangement just like a normal functional gene segment, a significant proportion of the rearrangements will incorporate a pseudogene and thus be non-functional. The immunoglobulin gene segments are organized into 3 clusters or genetic loci: the kappa, lambda, and heavy chain loci. These are on different chromosomes and each is organized slightly differently for humans. At the lambda light chain locus, located on chromosome 22, a cluster of V lambda gene segments is followed by 4 sets of J lambda gene segments, each linked to a C lambda gene. In the kappa light chain locus, on chromosome 2, the cluster of V kappa gene segments is followed by a cluster of J kappa gene segments and then by a single C kappa gene. The organization of the heavy chain locus on chromosome 14 resembles that of the kappa locus, with separate clusters of VH, DH, and JH gene segments and of CH genes. The heavy chain locus differs in one important way. Instead of a single C region, it contains a series of C regions arrayed one after the other, each of which corresponds to a different isotype. Generally, a cell expresses only one at a time, beginning with immunoglobulin M. The expression of other isotypes, such as immunoglobulin G, can occur through isotype switching. And that is, there are multiple different V region gene segments. Have a great day.